All right, welcome to Chapter 3. This is the revised version of the slide set, so I welcome you. Let's begin our discussion about cells. Cells, of course, make up the smallest portion of who we are, and the sorts of what we're composed of individually. All 100 trillion of these we can find throughout our bodies, and of course the cells make up all other living things. So in this section we're going to describe what a cell is and the two general types of cells. We're going to describe the structure and functions of the cell membranes. We're going to describe several ways in which molecules move across membranes, describe how cells are connected and how they communicate, and also 9 important landmarks of the eukaryotic cell we're going to discuss in somewhat detail. So what is a cell? Of course, all organisms are made of cells, as I was mentioning before. The cell, being the smallest unit of life, of course, can function independently and perform all the necessary functions of life as a commune, or as each part contributing to the greater whole, and which these functions, of course, include reproduction itself, or carrying new copies forward. So, where do we begin if we want to understand how organisms work? Well, given the complexity, this task can be daunting. Fortunately, as with most complex things, a strategy of divide and conquer comes in quite handy. In the case of organisms, whether we are studying a creature or a small flea, or as large as an elephant or a sequoia tree, they can be broken down into smaller units, and so more easily studied and understood. And that's what we're going to try to do, at least during this session. The most basic unit of any organism, of course, is the cell, the smallest unit of life that can function independently and perform the necessary functions of life, and including reproducing itself. Understanding cell structure and function is the basis of how complex organisms are organized. So if you follow me, we'll continue on. And the best description, I guess, is utilizing a microscope we talked about in the laboratory. Robert Hooke, who was a British scientist in the mid-1600s, he was able to describe and actually publish works on these microorganisms in a publication that he had. And this was a landmark from the standpoint that he was the first to describe and publish the work about what he saw is subcellular, using a microscope, which are beyond what we can see with our standard eyesight. So he described these and drew his images that he saw within the microscope, and this was actually described in his work. A cell is a three-dimensional structure like a fluid-filled balloon in which many of the essential chemical reactions of life take place. Nearly all cells contain DNA, which is the component now we're going to talk about at great length at various other segments during this presentation and other chapters that we're going to cover. Deoxyribonucleic acid is a very dynamic molecule, and it encodes all the instructions. It's the recipe set for who we are, or what our cells do, when things are done, and all of the steps in between. It's very important that we go over what DNA is really all about. So, of course, the term cell was first used by Robert Hooke when he was utilizing the microscope, and he took views of cork, which we know what cork is, cork boards and that sort of thing, And looking at the celluli or cellular look, small rooms, these empty rooms that he saw within this cork, and it was quite an interesting thing because you couldn't see this with your plain eyesight, but if you go in and zoom in, you could see these little rooms, and celluli is what he called these, and that hence became cell. So also looking at nearly all the cells, of course, that contain DNA, a molecule that contains the information that directs the cell's chemistry and the like. All of these things are important to make up the cell. Of course, he couldn't see these components, the DNA and all the other aspects that we're talking about here, but this was the step in the right direction. So we're going to explore more detail about these cell functions as we go into the following chapters. So how to see a cell? Well, it depends on the size of the cell. Hummingbird egg is about 0.02 ounces. An elephant bird, extinct egg, is about 2 gallon size. Beluga sturgeon eggs is about $700 per ounce versus a human egg is a couple $1,000s per egg. So value and size are shown here. So throughout, we're going to be utilizing some vocabulary in talking about pieces and parts of the cell, and this is a really good way to study, is to get your brain wrapped, I guess is the best way to say it, around the terminology of the various things. And I've always tried to emphasize that in class, and I try to provide tools, study tools for you to get to know the vocabulary quite well. So if you find on Blackboard the access to the vocabulary for Chapter 3, then I think you'll be well on your way. You can print that out or just follow it and make sure you kind of check them off as we go through, that you understand and know these terms, and you'll see that the exam performance will improve. So the cell, which is the basic structural, functional, biological unit of all living organisms, cells are the smallest unit of life that is classified as a living thing. And we often, they're called the building blocks of life. But let me go through this key, and if you follow the numbering and look for the various structures, the nucleolus, the nucleus, the ribosome, which are the little dots that you see in the various structures, which make up the rough ER, which is number 5, the Golgi apparatus, or you might see it listed as Golgi body, the cytoskeleton, which it comprises components of these fibrinous materials. It's sort of like the scaffolding you might see with folks painting or doing work. It's sort of the infrastructure. It gives the strength for compression and the like. We have the smooth endoplasmic reticulum. This doesn't have as many of the ribosome dots as you see as the rough endoplasmic reticulum does. And a little bit more fine processing, glucose and other types of additional molecules are added to finish up, and it's the sort of the finishing or the spiff and polish area there that moves on to that goes on to the shipping department, which we're going to talk about in a little bit. But the shipping department, of course, is the Golgi apparatus. The mitochondria are the powerhouses. These are the generators, the power generators that are required, and you can have more than one in a cell depending on the type of activities and the requirements for forming energy or requiring the use of energy. The vacuole, 10, are storage facilities. Could be fats or oils or water, various things. The cytosol, of course, is the fluid that makes up everything that's inside the cell that gives it the stuff that that the organelles and the like are bathed and constantly in. But that's where all the nutrients, we say nutrients, but the building blocks for building the proteins, the amino acids, and all the other things that are required for the building blocks can be found there. Lysosome, which is the shredding or the shredder. Lyso means break up, some means body, so it's the breakup body, or it's a container that disposes of things. The centrosome, which is sort of like the forklifts, but it moves things that very much like the fibers that you find in muscles. These all they can do is contract, but they move things like chromosomes and various other things that have to be moved. And of course, number 14 is the cell membrane. So I think we've gone over at least some of the basic parts that we're going to be using or discussing throughout this presentation. So you might want to keep an image of that close by just so that you can see this. So again, just for review, cell theory is all living organisms are made up of one or more cells. And if you recall from chapter 1, a theory is true until proven otherwise, and it's pretty much past the test of time. All cells arise from other pre-existing cells. And so if you're asked about cell theory, of course, and maybe even as a short answer or something like that, just refer to cell theory as a scientific theory that describes the properties of cells. And it's the basic structure in all living things. The initial development of the theory, though, was in about the mid-17th century, was made possible, of course, by advances in microscopy. And you can go back at the early part of the presentation and refresh yourself. But a prokaryote is a very simple cell. Just to review here, it looks like a rubber band that's been snarled up, and it doesn't have any specialized structure, sort of what we saw in the eukaryotic cell with the membrane and the like. This is just floating around in the cytoplasm. We have cell membrane, or the plasma membrane, the cell wall, and the capsule. The capsule is really there's, if you're a Star Trek fan, a cloaking device, but it's a polysaccharide that tries to fool the system if it does invade in humans, or it helps provide resistance against drying up or desiccation. Flagella is something that the bacteria use to move. It actually spins in two directions. One, it moves forward, and another rotation, it starts to flip and flop around until it finds some better environment. So the prokaryotes are simple, and eukaryotes are the more complex and make up cells like ours. If you can remember, that would be great. So, in summary, the most basic unit of any organism is the cell, the smallest unit of life that can function independently and perform all the necessary functions of life, including reproducing itself. All living organisms are made up of one or more cells, and all cells arise from other pre-existing cells. So, every cell on Earth falls into one of the two basic categories: eukaryotic cell, which we just discussed or started to discuss, and then the prokaryote cell. So the eukaryotic cell has a central control structure, the nucleus, which contains its own cell's DNA, but it's within a container, and it's like a huge cubicle, but it's isolated from the rest of the cytoplasm, although it's in the cytoplasm and communicates and shares things with it. It's isolated, and it's for a higher level of purposes for that. Where prokaryotes don't have a nucleus, and its DNA simply resides in the middle of the cell. So I hope you can remember that. And here's some simple drawings of what I showed before. But the idea with this is you can see all the compartments and the components, the special cubicles, if you want, where a prokaryote, everything is sort of communal all together without these compartments. So again, I just wanted to make sure that we have that. So if you look at the vocabulary that was in your study guide, the more defined definition is it's an organism whose cells contain a nucleus and other structures. And organelles is an important word to denote, because those are specialized offices that contain or provide a function that's unique. but they're enclosed within membranes, and that's the deal. I want you to remember is that organelles, of course, have their own membranes that isolate themselves from the rest of the cytoplasm. Eukaryotes are formally the taxon Eukarya or Eukaryota, and the defining membrane-bound structure that sets eukaryotic cells apart from the prokaryotic cells is the nucleus, or having that nuclear envelope or that cubicle that sets apart from the rest of the cytoplasm. And of course, prokaryotic cells, the nucleus is not contained inside its own membrane. It's in the cytoplasm in general. So the presence of the nucleus in eukaryotes, hence their name, comes from good nut. So it's a good kernel, or a just being U is the good part, and karyon is the nut or kernel or cell, is what we become to know it now. So most eukaryotic cells also contain other membrane-bound organelles like the mitochondria we talked about in the Golgi apparatus, the shipping department. In addition, plants also contain chloroplasts, which they would be eukaryotic. Many unicellular organisms are eukaryotes, such as protozoa, and all multiple cellular organisms are eukaryotes, including animal, plants, and fungi. And not to leave the fungi out. It's not fungi, that's somebody you want to invite to a party. A fungi is the right way I would like you to say that word there. So it makes it quite clear the demarcations here. If it's got an isolated or its own membrane, then it's a eukaryote. Prokaryotes are a group of organisms that lack cell membrane and that carry on that we talked about. The organisms whose cells do have a nucleus are called eukaryotes. The prokaryotes are unicellular organisms. So try not to get these two confused. I try to keep these straight and simple. Prokaryotic cells, of course, like the mitochondria and chloroplasts, and they have processes such as oxidative phosphorylation and photosynthesis that take across the prokaryotic cell membrane, the large cell itself, but they don't possess those internal structures like the eukaryotes do. So both eukaryotes and prokaryotes contain RNA and protein structures called ribosomes, which produce protein. Prokaryotic cells are usually much smaller than eukaryotic cells. Therefore, prokaryotes have a large surface to area volume ratio, giving them the higher metabolic rate, higher growth rates, and as a consequence of that is a shorter generation time than in eukaryotes. So prokaryotes really can grow about 20 minutes or so as a generation time. In other words, they can divide every 20 minutes. Think about that every time you leave a potato salad outside, and come back later and see if how well you feel in about 3 hours. So here's a more detailed out of your book in figure 3.3. If you look at the prokaryotic basic structure, there's that rubber band I was talking about. That's the chromosome, and there are some extra chromosomal inheritant pieces that we call plasmids, or recommended or diagrammed here. We also have the flagellum, the pili, which are hair-like projections that help the cells attach. The cell wall gives it protection. This doesn't have that polysaccharide coat, and it's not shown here, but we have already identified. There's the ribosomes that both prokaryotes and eukaryotes have, but you notice it's lacking all those cubicles, or those membrane organelles inside. That's, again, a very distinguishing feature between that and a eukaryote. So in summary, every cell on Earth is either a eukaryote or a prokaryote. Prokaryotes, which have no nucleus, were the first cells on Earth. And we'll continue our summary with 3.2, that they are all single-celled organisms. Prokaryotes include the bacteria and archaea, the old ones. These are they've been around for a long time, as during the development of the planet, and just think of the archaic or old might be a good way to remember that. Archaea, as a group, are characterized tremendous metabolic diversity. They can grow in very hot climates or environments like thermal vents at the bottom of the oceans to very cooler, much cooler temperatures because of how they existed early on, a long time ago. So eukaryotic cells have compartments with specialized functions. You can see all the specialization going on in the different climates or environments that they live. And eukaryotes have organelles, and we've talked about the organelles many times already in this presentation, but just to redundancy leads to good memory production. So eukaryotes have a true nucleus that's contained within a compartment. Internal organized structures or compartments we refer to as organelles. They're larger than prokaryotes and usually about 10 times bigger. And the cytoplasm contains specialized structures that we've already referred to as organelles, where the prokaryotes don't. No nucleus for the DNA is just loosely associated within the cytoplasm, but it's circular like a rubber band. Internal structures not organized into compartments. We don't have organelles, and they're much smaller. So there are some differences between the two, and you can find this in your book with figure 3.5. And so if I were to look at some differences now within eukaryotes themselves, the animal and plant cells, then you can see pretty much everything is the same except how we contain it. And everyone I guess has seen plants wilt. Well, we have some specialized structures because plants use a different type of vascular system than perhaps other forms of life do. They have these rather large vacuoles that contain water that make the plant more turgid. In other words, they're not wilting, they're straight up and they're strong. But if you lose water, then we have this specialized structure in the plants that we don't see in the animal. Cell wall we can see is listed here, and that's much more rigid because these are outside and they have to weather the various components, and they can't just like walk to a shelter or something. So they're a little bit more robust, and we have a cell wall where the animals don't. And then chloroplasts are these photosynthetic or solar cells, I guess is the best way. We're going to talk about photosynthesis soon. A later chapter here, in chapters 4. But the chloroplasts are really interesting because they capture energy from the light and convert it to sugar, and then that sugar is used for storage for energy, and then the sugar can be converted to a very useful component called adenosine triphosphate, or ATP, which is the universal currency or the energy that can be utilized throughout a cell. So not only plant cells but animal cells use the same. So these are the structures that are found in both. Structures not found in a plant cell is the centriole. These move the chromosomes and other components, and the cell plants, the cells in plants, have a different way of dividing their DNA, and so they don't need the forklifts like those do. So which feature below is only found in eukaryotic cells? And if you think about this, it's not meant to be tricky, but if you think about this, then the answer would be mitochondria, because both prokaryotes and eukaryotes contain ribosomes, DNA, and cell membrane, but it doesn't say or reflect too much about its components. But mitochondria are not found in the bacteria cells, and of course this is a membranous bound organelle, so that should have been some clue for you as to what is the right answer. Endosymbiosis. Endo means within, symbo or together life. So an osis is a condition of together life within. So it's life that's sort of grown together. So that it's a theory that's been developed to explain the presence of two organelles in eukaryotes: the chloroplasts in plants and algae, and the mitochondria in plants and animals. These are the energy components of these two types. So, endocytosis is an energy used to process which cells absorb molecules such as proteins by engulfing them, and is used by cells in the body because most substances are important to them. And this is a way that cells move in and out. I just wanted to make reference to this because we're going to be talking about endocytosis and exocytosis before too long. Anyhow, this endosymbiosis is the ability to bring in other types of cells. And sometimes you might hear this endocytosis, not to be confused with the endosymbiosis theory. So although the endosymbiosis theory means that the cells had to be moved inside, it's not as a result of just endocytosis. Okay, so it's life that's merged. So humans deep down may be part bacteria. How can this be? Well, we're talking about this endosymbiosis. According to that theory, two different types of prokaryotes may have come to close partnerships with each other. And for example, some small prokaryotes capable of performing either photosynthesis or having the energy like utilization like the mitochondria, these cells have come to live inside the larger host prokaryote, or in our cells, in eukaryotes. So the mitochondria and the earlier forms of life have adapted, or these forms of life have come together in our host as well. So a better way to see this is with this diagram here. So endosymbiosis, we have, of course, this ancestral prokaryote, and going through this process, and this is that ability to invaginate and to merge these sorts of things with this endocytosis sort of process. And invagination is a process where the plasma moves in, and we get inner compartments, organelles, formed as a process of this invagination. It could be as a result of this endocytosis process, but it's something to think about, and I just wanted you to be aware of that terminology. Now, to throw some text at you, and this is in your book in Figure 3.7, but I just wanted to be complete, and of course to make sure that we cover some of these key words. You should become familiar with these words as you work in your textbook, and highlight these sorts of words so that you get more and more familiar with the vocabulary. So two cells that may have become more independent from each other could not live without each other. They became single, more complex. So the prokaryote evolved to be a chloroplast, an organelle that the plant cells have taken in, and then some prokaryotes may have evolved into a mitochondria, and that organelle has been able to be taken in. So mitochondria versus mitochondrion, this is a singular form, mitochondria is a plural form. So how do eukaryotic cells become so structurally complex? Of course, is this process of the endosymbiosis is supported by these observations that I've listed here. Chloroplasts and mitochondria are similar in size to prokaryotic cells. Chloroplasts and mitochondria have small amounts of circular DNA, similar to the organization I've found that we talked about prokaryotes. The chloroplasts and mitochondria divide by splitting, just like prokaryotes. And chloroplasts and mitochondria have internal structures called ribosomes, which are similar and found between the two. So they use the same sort of mechanisms to make proteins and all the other things. So it's very compatible. So in addition to the theory of endosymbiosis as an explanation for the existence of chloroplasts and mitochondria, another theory about the origin of organelles and eukaryotes is the idea that the plasma membrane around the cells may have enfolded upon itself a process called invagination to create inner compartments. We just don't know which is the correct way, but the DNA that's contained is different from the chromosome DNA. So the mitochondria and the chloroplast DNA is definitely not from our own tissues. So I do want you to know the endosymbiosis theory, and I've given a textbook kind of definition here, but you can draw a diagram of various things similar to the figure in your book, and the endomembrane system to be familiar with these sorts of things, and your diagrams. Of course, these can be found in your vocabulary list in addition to this set of slides here, but I do want you to know these words. So let's do a practice question here, and so that just to make sure that we're on the same sheet of music. And which evidence below supports the theory that mitochondria and chloroplasts were originally bacteria? And so I'll give you a few moments here to think about this. And I look at some of these answers here. Both organelles are larger than the other organelles in the cell. Well, we know that's not true. So that kind of rules out 2 and 4, because all of the above and 2 are obviously not compatible. Both organelles are surrounded by a single lipid bilayer. Well, that's not true either. So I hope, anyhow, that you came to the conclusion that circular DNA is present in both organelles, which are more consistent with prokaryotes, and I think that is a good answer there. So in summary, eukaryotes are single-celled or multicellular organisms whose cells have a nucleus that contain linear strands of genetic material. They are also commonly have organelles throughout their cytoplasm. So having organelles which are phospholipid bilayers, which may have originated evolutionary through embocytosis or invagination. I tend to lean towards symbiosis. I'm curious to what you think, but anyhow, it seems very plausible. So I'm going to stop here for this section and follow it up with section 2 for our enjoyment and pleasures for later. So I appreciate your time, and you can review this section, and I would advise just going through the laboratory, the dictionary and the various things there, so that we can go over this at a later time on an exam and do quite well. So I'm hoping that everyone will perform well in the exam if you study. So let's go ahead and wrap it up today, and you can look for section 2 here in a little bit. Thank you.